there? Oh,
I know my family finally brought him back, so I'm
what a
finally able to see him.
beauty. Oh, he's a lovely one, he is, that's for sure. All right, well, welcome to the next section. So today we're going to, let me just get everything up and running. Go. Right, so essentially this section is, on Jesus, mostly about gene expression, central dogma type stuff, right? So we're going to start off with a look at eukaryotic chromosome structure and dynamics, right? Because that's a real important part of control of gene expression. And it's also kind of an interesting aspect of our biology, essentially, as well. It's something that not many people are really aware of, I think, in terms of how much kind of empty space, not empty space, but how much kind of evolutionary cruft we have in our genome. It's kind of like thinking about space, you know, it's very big, got lots of planets and stuff in it, but they're very spread far apart and there's nothing much in between them, right? So it's the same kind of thing in terms of our genome, which is a little bit weird to think about. So that's really today, mostly today, a little bit on Thursday. Let's see, oh, I got to close some of these documents. And then on Thursday, we're going to get kind of dive straight into gene expression and transcription, right? And so the reason why we're going to spend so much time, like the entire section pretty much, on this is because gene expression is really, you know, underpins all biology, right? It's not, you can't really get around that, you know, the way in which we inherit our information is through DNA by and large. And the way we turn that information into something useful, right? Because DNA in and of itself is pretty useless, right? It doesn't do anything. The way we turn into something useful is through the process of gene expression, right? And so the differences between you and I, right? Or, you know, you and your dog next to you, or, you know, you and your gut bacteria, well, that's like a little bit more of an extreme stretch, isn't just what DNA you have, although that is part of it. It's also how you turn the information in that DNA into a functional product, right? And so to be able to understand really like the enormity of biological form and function, you really have to understand the basics of how information is turned into function, right? And so that's really what we're going to be spending pretty much the rest of the course on, right? So there's going to be a lot of detail type stuff, right? So you've had a kind of a exposure to that from the last exam. Exams 3 and 4 are going to be even more of that, right? So there's a lot of mechanisms, lots of processes, lots of things to remember. And you can't really have one without the other, right? So you have to understand the general processes, you know, how that all fits into the big picture, how things work. But you also need the details, because otherwise that big picture doesn't make any sense, right? It's like if you skim read an article in a newspaper, you know, you might have a general idea of what's going on, but you're not really going to be able to explain it to anybody else or use that information in a different context, right? And so that's really what we're going to be working on for the rest of the semester. And if we have time, hopefully we will, at the end, we'll look at some fun stuff with transgenics and biotechnology, right? How you can apply those things to that knowledge or those skills or whatever to kind of, you know, real world type stuff. So anyway, what we're going to do today is we're going to rip through this PowerPoint and try and get through as much of transposons as possible. And I always like talking about transposons, partly because I actually did some research on one called Piggyback, which is kind of a cute name. Many moons ago when I lived in, shoot, where was it? Australia, I think. Can't remember. Something like that. And also just because they're really, really neat. They're kind of very curious little things. Can't call them creatures, but they're kind of like DNA parasites in a sense. You know, it's kind of weird. Anyway, so we'll start on that at least today. Whether we'll finish it or not depends on how things go and how distracted I get. Okay, so essentially, this is all about eukaryotes. You know, prokaryotes have their own quirks in terms of genomes and stuff, but eukaryotes are far more complex, both in terms of their genomes, in terms of their genome dynamics, and obviously in terms of their biology, right? You know, you don't have any, well, yeah, bacteria do funky, cool stuff, right? You know, they talk to each other and stuff like that. They live in communities, but they're not multicellular organisms like we are. So you kind of have that bit of a smug feeling about we are complexes of cells rather than just cells. Anyway, so this is I'm guessing this is what's called a fish staining. It doesn't use fish. It's short for fluorescence in situ hybridization. And so basically you're tagging bits of DNA with different colors, fluorophores, essentially. We'll see a little bit of that later on, to identify different bits of DNA, and also makes for a pretty picture as well, which is no bad thing. And essentially this is a picture of some human male, because there's a Y chromosome, chromosomes during cell division. My guess would be during mitosis and prophase or thereabouts of mitosis. And you can tell, right? So I'm not just blowing smoke up your backsides, but you can tell because, let's look at this one over here. Very faintly, you can see the line down the middle. That's the gap between the sister chromatids, right? Also, because, come on, back you go. These chromosomes are condensed, heavily condensed. You only ever see that during cell division. Partly because you have to, right? So, you know, it's like, I always use the analogy, it's the difference between serving spaghetti and serving meatballs. You know, it's a lot easier to count out meatballs than it is strands of spaghetti. And that's essentially the same deal during cell division, right? Condensing them makes them a lot easier to segregate, essentially, and organize, you know, that sort of stuff. But also, I know this is cell division because these being condensed, these chromosomes are transcriptionally inactive. They are shut down. There is no gene expression whatsoever occurring from these chromosomes, right? Because all of the various machinery, which we'll cover soon, cannot access the DNA, right? So essentially that's why all of the things required for cell division, like all the way through, are made in interphase. Because as soon as that cell enters M phase and these chromosomes condense, you're not making any more of anything, right? You're pretty much committed and you have to hope you have everything you need. Okay, so anyway, also you can see these are lined up by size, right? So these are sex chromosomes over here. We've got a lovely, fabulous looking X chromosome and a stunted little midget of a Y chromosome. So in some senses, we are genetically inferior to females, guys, sorry. I'm sure all the ladies in the audience like, yeah. Anyway, and also these are organized by size, right? So chromosome 1 is chromosome 1 simply because it's the biggest, right? No other reason, right? And so you can see there's a large difference in size of these chromosomes, right? And we'll get to that later. Now, something that's always struck me as, pretty curious, right? And obviously likewise to other people, is that there is no rhyme or reason behind the size of a eukaryotic genome. So we have, oh, what was Sarcodina? Oh, these are amoeba. So Sarcodina, I don't know that before yesterday, are amoeba and other kind of stuff, single cellular eukaryotes. And they have, some of them, the biggest genomes of all eukaryotes, right? Single cell organisms, right? And so clearly in terms of organismal complexity, they're fairly way down the list. Yet they have, some of them at least, the largest genomes that exist, right? Actually, quick aside, because it's just fun. I'll have to see if I can find a picture of this sometime. But a guy that I used to know from previous job does a lot of deep sea biology. And in one of his cruises, he or the people he was working with found an amoeba that was about that big. So single cell amoeba, bottom of the ocean, I don't know, 4,000 meters depth or something like that. And it had a radioactive crust. So it actually coated it somehow itself with radioactive minerals from its environment. Absolutely, totally bizarre. Obviously, they can't study it because, you don't want to, it's going to be hard to pick up because it's an amoeba, but also it's radioactive. So anyway, that was kind of like a funny thing to think about. And we're, more or less in the middle, give or take. So genome size has nothing to do with organismal complexity or phylogenetic kind of relationships, so we don't necessarily have more DNA than things we think are less evolved than us, which is always a little bit of a difficult, term to use. Also doesn't have anything to do with gene count. So I don't know where. Well, I imagine that, what I work on, C. elegans, is somewhere in here. It's not an annelid, it's a nematode, but it's somewhere down here in terms of phylogenetic order, I guess. It has exactly the same number of genes, give or take, a few hundred, as we do. So this is an organism with, in the hermaphrodite form at least, 989 cells exactly, and people know because they have counted them. And kind of lives off of bacteria and rotting fruit and stuff like that. And it has exactly, we'll give or take, roughly the same number of genes as we do, right? And we clearly don't have 989 cells or live on rotting fruit in compost. So really, there is no particular correlation between any of this and genome size or gene count. And this is actually called the C-value enigma or C-value paradox, where the C-value is the DNA content of the haploid genome, right? And so we use the haploid genome because that's like the unique set of information. Essentially, we have 2 copies of it, which are largely the same, but slightly different. Right, so that's why we use the haploid genome. So in terms of actual, amount of DNA, we have about 6 times 10 to the 9 base pairs, right, or 6 gigabases of DNA in our somatic cells, right, our non-germ cells. But we never really use that. Kind of reuse the haploid genome. Oh, also, so anyway, this is the enigma or paradox, and really what it represents is kind of like the evolutionary history of that organism. So for example, down here, I don't know where wheat fits in. Gymnosperms, maybe? I don't know. I'm a really shitty taxonomist, to be honest, and I have no worries about that at all. But wheat, for example, the stuff that you make, you know, tortillas or arepas or anything like that out of bread, you know, is the result of 2 rounds of genome duplication, right? So wheat is hexaploid, right? It has 6 copies of each chromosome. We are diploid, we have 2, right? Same thing happened with maize, you know, also used to make tortillas. That originally came from a plant called teosinte, which you can see alongside the roads, you know, in San Antonio and certainly further south that you get towards Mexico. You know, it's kind of like this weird, like green bush type thing with like these little corn cobs, essentially, right? And that, through, again, a couple of rounds of genome duplication, resulted in what we now know as maize, right? And a whole bunch of selection from that point onwards, right? So, you know, they have huge genomes, but it's not, or they have huge kind of DNA counts in their cells, but it's not because they're more complex, it's just because of what happened in their evolution, essentially. And so in many ways, the size of our genome reflects the amount of DNA baggage that we carry with us. Essentially, DNA that is not deleterious, right? It doesn't cost us anything in terms of fitness, you know, upon which natural selection acts. It might not actually give us any benefit either, but it doesn't harm us. So essentially, we just carry it around generations and generations. There's no real way of losing it unless by selection. And if it's not deleterious, then we're not going to get rid of it. And so one big reason for all of that cruft, right? So essentially, I don't know what it would be, probably about 3 to 5% of our genome equals protein coding genes or regions. Right? So a very, very, very small amount of our genome actually contains information used for making proteins, right? Really small amount if you think about it. I mean, that's a large part of what makes us what we are, right? Some more, and that's unknown, equals coding genes, e.g. Non-coding RNAs. You know, so an example for one of those would be ribosomal RNA, for example. You know, the molecule that's involved in translation doesn't produce protein. Well, I mean, its actions produce proteins, but it is not used to make a protein as information. Yeah, hopefully that isn't confusing. And we don't know how much of our genome is used for that because they're really, really hard genes to find, right? They don't have the characteristics of protein coding genes. So you can't find them, you know, bioinformatically. You have to do it experimentally, which is very slow and random, essentially. So all of the rest, which is the vast majority, right? Say 90%, maybe more, of our genome is made up of repetitive sequences and satellite DNA. And old defunct genes, right? So genes which no longer function. They're called pseudogenes because they're not real genes. You know, stuff like that, evolutionary cruft. And these sequences are typically repeated over and over and over again, and as many as 1 million times, which is kind of crazy. And these sequences are called satellite DNA. right? And so a large part of the difference in genome size between organisms is down to how much of this stuff they have. Now, these repeat sequences may be, well, there are lots of different types essentially, right? They could be single copy sequences, right? So old viruses, right? For example, integrative viruses that are no longer active. HIV, for example, is a virus that integrates into your genome, as is shingles, varicella. What else? There are a few other odd, viruses and the like that kind of hide in your cells. Some of them are transposons as well, which we'll talk about later. Some of them are, and that's kind of what would be, what these would consist of, these single copy to middle repetitive sequences. The highly repetitive sequences, of which there, is a huge amount, up to almost half of your genome, are small repeats, repeated over and over and over and over again. And one of the reasons for that is that DNA polymerase, the enzyme that copies your DNA, tends not to work very well on repeat sequences. So it does something called stuttering. It kind of slips. So DNA replication. Oh, give me a sec, Kiana. I'll get there in a moment. What would be the best way to describe stuttering? Basically, where it slips, stutters on repeats. And a good example of that actually is Huntington's disease, which is called a poly-Q disease. And that's because the DNA polymerase, when it replicates, adds those repeats by accident, essentially. And because those repeats are in a coding gene, that causes problems, but it also does the same elsewhere. So, Kiana, good question. Basically, it represents the diversity of the various different things you have within your genome. So we have a lot of different things in our genome that may only be present once or a few times. That's what these categories kind of represent. But we also have potentially a large chunk of our genome, 45% is pretty large, that is repeated over and over and over and over and over again. And so the reason why the lower bound number gets smaller is because it depends on what organism we're looking at. And so essentially, every organism has to have all of the genes it needs, right, to be functional, and all the other stuff it needs to be functional. So there's kind of like a minimum genome size, essentially, right? Where if you get smaller than that, you start giving up function. And you can see that in various parasites, and particularly kind of pathogenic bacteria have this, where they have very, very small genomes because they don't need as much stuff. And so essentially the rest of the difference between, say, us and, I don't know, wheat or, pufferfish or whatever, right? The rest of that difference is largely made up of these highly repetitive sequences. And so that's why the number gets smaller, because in some organisms, you have a very small amount of highly repetitive sequences, and they have a fairly small genome. Other organisms have huge genomes, and the reason for that is largely because they have lots of this stuff. So that's the kind of general arm waving kind of answer I guess. Does that help? Cool, cool. And so also there's, not only is there big variation between organisms, there's actually a really huge variation between chromosomes, right, in a given organism, right? So chromosomes are not uniform structures or entities, right? Either between them or even within them, which we'll get to a little bit later on. And so basically they vary hugely on size, right? From 280 megabases, so that's million bases, all the way down to the Y chromosome, little stunted chap, which is 128 megabases, right? So there's a large difference in size there. Actually, sorry, I got the wrong size. It's 50 megabases, right? So 21, 22, and Y are both, really itty bitty. But just look at the difference between 22 and Y. 22 has nearly 800 genes. The Y chromosome has 128. Part of the reason being is that those Y chromosome genes are largely, male specific genes. Because they're not necessary for female development, because otherwise, obviously there would be issues there. But even between 21 and 22, more or less exactly the same size, the gene density is different by over a factor of 2. So again, that kind of represents essentially what those chromosomes kind of were created from in terms of gene duplication, chromosome duplication, chromosome merging or splitting. There's all kinds of kind of weird things that happened in our evolutionary history. And similarly, we've got a huge chromosome up here, has the same gene density as our Y chromosome, right? So there's a lot of variation. And even when you look at it in terms of our individual chromosomes, right, there's variation in gene density among the different parts of that chromosome, right? So gene density is pretty high in the arms, right? Whereas generally DNA generally takes the form of euchromatin. So euchromatin basically equals open transcriptionally, if I can spell it, active DNA, usually on the arms of the chromosome, right? Whereas near the centromere, and the centromere doesn't always have to be actually at the center of the chromosome. Some different organisms have it way up near one end for some reason. But the centromere and the telomeres are typically heterochromatin. Transcriptionally silenced, always at the centromere and telomeres. Sometimes it depends basically on the arms. So the arms can also be transcriptionally repressed, but the centromere or centromere and the telomeres are always shut down. And there's a lot of kind of non-coding DNA in those regions. In fact, the telomeres are entirely non-coding, right? For good reason, right? You don't want any genes in those. They're there to act as buffers. It's just a whole bunch of repeats. And we'll be getting into euchromatin and heterochromatin a great deal more when we look at control of gene expression in eukaryotes. Okay, any questions so far?
What do you mean when it's when you say it's closed transcriptionally?
So closed as in physically. It's actually more heavily coiled and the DNA is less accessible. So let me write that out. So it's, oh, if I can spell that right, typing sucks today. And yeah, it's physically actually more tightly wound, right? And so there's different levels to this. You could be looking at the ultrastructure of DNA, or you could be looking at the actual interaction between DNA and the histones that form chromatin. But either way, that DNA is less accessible. And the upshot of that is it's not available for gene expression. It's basically tucked away. You know, it's even if the various factors that would turn on the expression of a gene were present, if the DNA is physically inaccessible, then it's not, that gene is not going to be transcribed. So it's not only is it a kind of a structural thing in terms of you know, what region of the chromosome, it's also a way of shutting down large parts of the chromosome. And so good example of this actually is X chromosome inactivation in women. So in Drosophila, which have an XXXY sex determination, the X in males is boosted. The expression from the X in males is twice that of females. So essentially, females and males have the same amount of expression from the X chromosome. In humans, however, we do it the other way around. Again, it's XXXY, but now in females, every cell has one of the X chromosomes inactivated. Which one it is totally random. That's why you can get like funky things like calico cats and maybe other phenotypes in humans. I don't know what, obviously not calico. But yeah, so that's the way how we manage, you know, dosage of the X chromosome in humans, is that in every single cell in a human woman, female, one of the X chromosomes is shut down. So no expression from it whatsoever. And so when we look at how that's done, so that's partly for packaging reasons, right? So DNA is physically, relative to the size of the cell, huge, right? There's literally a lot of it, right? And so we have to have some way of kind of stuffing it all into the nucleus. It's like basically, how do you pack your suitcase kind of deal, right? But it also has to still be accessible, right? Because we need that DNA to be able to make mRNA and proteins, or just RNA. So part of that's done by supercoiling the DNA. So if you remember from DNA replication, topoisomerases can relieve supercoiling. They can also introduce it as well. But most of the compaction of the genome is done by the formation of chromatin. And so chromatin is DNA and protein. And that's why in the Drosophila, the DNA isolation lab last couple of weeks, you added proteinase K to your samples. And the reason why you did that was to degrade the protein that was complex with your DNA, or the fly DNA that you're trying to extract. Because you don't want it there. It just gets in the way of, PCR, and, actually interferes with its binding to the mini column as well. Right. So that's why you had to do that digest, is to get rid of that protein. And essentially there are 5 different types of histone proteins. You have 4 pairs. They're called H2A, B, 3, and 4. So you have 8 proteins total. Kind of makes the ball around which the DNA is wrapped. It's kind of wrapped 2 and a bit turns around it. 2.4 turns, I think. And you have a 9th protein called the histone H1 protein. And so that kind of functions to hold everything together, and it also helps space these balls apart. Let me just add some language too, right? So we have two different terms here, and they're kind of important to differentiate between them. So we have the nucleosome. That basically equals the DNA and proteins from the start of one of these core particles to the start of the next one. And that is, around 200 base pairs DNA, give or take. 8 histone core proteins and 1 histone H1. And when you look at them, in kind of bigger plan, basically the DNA or chromatin looks like so, right? These beads are spaced somewhat apart. And part of the reason for that is that it allows those kind of structures to kind of rotate and twist around themselves. So it kind of gives a little bit of flexibility. Right? You don't want them packed right up next to each other, because then it's much more of a rigid or inflexible structure. And part of it also is because this space allows you to move these histones or core particles up and down the DNA. Kind of like an abacus, if you think about it. So we can alter the DNA that's available simply by kind of, like pushing them to one side. So that's a nucleosome. Now, a core particle, or nucleosome core particle, is just the DNA wrapped around the 8 histones. And that's around 145 to 147 base pairs. So depending on what we're talking about, obviously changes a little bit kind of what we're referring to. Any questions? All right, so just before we move on, because this is actually a really important thing to understand, this DNA is attracted to the histones, not because the histones are, sexy, but because of the properties of the histones. What charge do you think the histones have? What charge? Think about what charge DNA has. Yeah, good stuff, Jacqueline and Lindsay. The histones are going to have a positive charge, right? Because DNA has a negative charge. So it's actually, and it's not shown here very well. Let's see if, actually, let's open up. Do. Ba, ba, ba. Give you a little bit of a fright. Ba, 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 ba. Yeah, go away. So actually what we have, this is a better one, is histones have tails. Kind of looks like some ugly blobby alien or something, I don't know what. But these tails actually stick outside of the core particle, and they're positively charged. They're what actually help hold the DNA tight. And so you can modify that charge to modify how tightly bound the DNA is to that histone. Right, so remember we were talking about heterochromatin versus euchromatin, heterochromatin being inaccessible DNA. Well, this is actually part of that, right? It's a way of kind of like mine, like those seagulls in Finding Nemo, mine, like really hugging onto the DNA. And so if you want to access the DNA, not only does the whole structure have to be open, but the DNA has to be available as well. There you go. I'm not going to look at any more of that because it's a bit of a scary PowerPoint. It's like 40 slides or something. So the other thing to, oh, and actually this is kind of a bit in the wrong place. Stick that up there. Actually, sorry. Before we get onto that, there are obviously many different levels of structure with DNA. And so all DNA, unless that DNA is being replicated in a eukaryotic cell, is going to look like anything from this to this, right? You don't have bare DNA typically in a eukaryotic cell. And so euchromatin would be this kind of 30 nanometer kind of thread, I guess. And heterochromatin is going to be more tightly wound and packaged, right? And so there's a very dynamic change between, for any given bit of a chromosome, between being shut down and being opened up. And so sometimes that part of the chromosome will be active when whatever function is provided by that DNA in terms of protein is no longer needed. It often gets shut down again, and that kind of creates a bit more space for other chromosomes to open up, to be transcribed, and then they get shut down. So it's a very dynamic process. It's not static in any way. And so just to finish this up, actually found this picture yesterday. So if you're looking at your PowerPoint wondering, where's this picture? This is new because I got a cool question yesterday. So I figured I'd go and actually have a look at it. And so if you think about, the nucleus as being a ball, like a soccer ball or a basketball or something like that, right? It's defined by the membrane, the nuclear membrane, which is a pretty interesting kind of structure. But inside of that membrane, right? Because it is like the skin of a ball, right? That is where chromosomes are attached, right? So each chromosome in each of your cells has its own distinct spot that it hangs out at. And it's actually physically attached to the inside of the nuclear membrane to kind of keep it in position. And so when you look at it in terms of individual chromosomes, It actually looks like this, where each of these colors is a distinct chromosome, right? So each has its own territory, essentially. And reason why I put this picture in is this actually shows you, this is an actual image of a nucleus. I think it's from a human fibroblast, right? So probably some kind of human cell line. And so they use all kinds of different fluorophores, and this is usually done, as I said, with fish. And so this isn't one picture. This is actually a kind of a stack, an overlay of all of the different pictures with false coloring, because they don't actually glow green. Well, they kind of do, but you don't take green pictures. You add color later on. And so you can actually see there's this, I don't know, well, I'm not sure what analogy I'd use for this. Badly stuffed sausage, maybe? I don't know. But anyway, each different spot, and you can actually, there's some really, really cool images. These are usually done using like a confocal laser microscope, which takes slices of your sample at, you know, different thicknesses. So that might be done on a, maybe like a 1 nanometer, no, 1 micron, sorry, 1 micron slice, maybe less. But you generate this 3D kind of image. So if you actually go to that paper, maybe, I don't know, in the supplemental materials, I'll actually have videos where you can just, you can kind of rotate the image around. It's really, really cool in 3 dimensions, essentially. And so when you look at it like that, now you have, you know, there's the X, there's the Y chromosome. Here are all the other different chromosomes in their own different spots. And as you can see, there's 2 chromosome 1s. One of them's over here, one of them's over there. So I think that's pretty cool. It's really weird to think about too. And it's odd as well to think about the fact that, you know, we always see representations of the cell and the nucleus as being kind of like these open spaces where there's, you know, these enzymes zipping around. And really it's a very tightly packed, highly ordered 3-dimensional space. So actually even thinking about, like, how on earth do you even find the right bit of DNA to transcribe? How does that mRNA get out of there? You know, if it was me, I'd just be wandering around in the nucleus forever. Like, because there are only a limited number of holes. It's not like this is a sponge ball. You know, it is actually kind of walled off from the rest of the cell. So anyway, well, that's pretty cool. And the paper is, yeah, down there. If you want to check it out, you're more than welcome to. Okay, right, before we skip on to transposons, questions? Nope. All right, so now transposons are actually really cool. And one of the reasons why I like them, obviously, as I said, is I worked on them for a while. Not very successfully, but I did work on them. And also because I'm a big fan of discoveries that kind of upend or overend biological dogma, right? So we like to think of ourselves as being a, you know, on the bleeding edge of science, you know, open-minded, kind of inquiry-based individuals. But actually, we're fairly well beholden to kind of existing dogma, how things work, right? And it's interesting because dogma is a very religious word in terms of, you know, canon, but it also applies in science. And so, you know, and that's actually one of the curious reasons why the central dogma is called the central dogma. Much to the regret of the people that called it that, Watson and Crick. But transposons broke another dogma, right? And so, when was this discovered? I think it was late 60s, early 70s. and it was discovered by a lady called Barbara, if I can spell her name right. Barbara? Oh, yeah, R in there. McClintock. And she discovered transposons in maize. And so essentially what she noticed is she's doing, breeding experiments and genetics of maize, right? She noticed that some kernels had like dark spots or light spots or different pigmentation anyway. And those spots, right, weren't inherited, or the underlying genetic cause of them at least, wasn't inherited in a classic Mendelian fashion. And so she spent a good while actually trying to figure out, like, what's going on? None of this stuff makes any sense, right? And she came to the conclusion that the only way in which this makes sense is if that phenotype is being caused by a piece of mobile DNA jumping around in the genome of that maize plant. And so that's why transposons are colloquially called jumping genes, right? Because they move around of their own accord. Now, she was widely panned. It doesn't help that she is a woman in science in that era either. But she was, almost ridiculed for this idea that DNA is not this static entity, right? Actually has parts of it that move around of their own volition, right? Without any kind of direction from the cell or something like that. And she was right. She kind of completely overturned that particular dogma. And in the end, she actually got a Nobel Prize for that discovery too, because it really was utterly groundbreaking, right? And since then, we've discovered, probably thousands of transposons, different types, that move in different ways and they exist in different organisms. And actually, there's a current thought on transposons is that they've actually generated a lot of new functions in our genome. So part of our evolution as organisms, part of the change in our DNA, which was selected by natural selection, was actually created by transposons. So that's actually pretty cool. Transposons are super neat. And so transposons are obviously, it's a concatenation of transposable element. So it's kind of stuck together. And they are found in all organisms, even bacteria, right? So all organisms that have, DNA genomes have transposons of one form or another. And they make up at least 45% of our genome. Right, so a big chunk of our genome consists of transposons. Not all of them are active, not all of them move around much, but some of them do. And so they can, they have their own very distinct mechanism of what's called transposition. And so transposition is insertion of a transposon to a new site. And there's a huge variety of them as well. In fact, until CRISPR-Cas9 came along, transposons, particularly the piggyback transposon, was the main way in which you created transgenic Drosophila. So it's a very heavily used tool for working on organisms. Now we have CRISPR-Cas9 and all that stuff is just like, whoosh, don't need that no more, because CRISPR-Cas9 is a lot more powerful, basically, right? And you can do a lot more stuff with it. But until, like 10 years ago, transposons were the way to go. That's why I was spending time on it. I was on a project to create a biocontrol agent for possums in New Zealand, believe it or not. I need to check, see how that's going. They're probably still just dropping, tons and tons of poison to kill them off. That's the usual way of doing it. People don't like that too much because if your dog eats it, it'll die pretty much instantly. Or a horse, or yeah, that's probably about it in New Zealand, or sheep. Anyway, so the general mechanism, this is a real weird kind of thing to think about, is that basically transposons recognize a particular sequence. In the genome that they live in or exist in. So e.g. piggyback recognizes any AATT. And so the thing that does stuff, I need to update this bit a little bit, is an enzyme called transposase. And that transposase is typically encoded by the transposon itself. So this yellow region in the middle contains a gene, and that gene is the transposase that it needs to move itself around. So that's why they're mobile genetic elements, because they contain the information to make the protein which is used to move them. And so that transposase, what it does, and obviously it depends on how long that target sequence is. So basically longer target sequences are rarer. Shorter ones are more common. So piggyback jumps around pretty much wherever. That's what made it a really useful tool. But whichever target sequence they recognize, what they do is when they find it, that transposase, the enzyme itself, it cuts the DNA. And it does what's called a staggered cut. So the transposase basically creates a double-stranded break, which we come across in homologous recombination. But this is very different. And it's staggered because it doesn't just cut the two strands straight, right? So it doesn't create a blunt cut, which is just like the two ends, like you would if you cut a piece of string. What it does is creates a staggered cut, so there's an overlap, right? These are actually called sticky ends, funny enough, right? And so that overlap is kind kind of key to the characteristics of transposons. Because once that cut has been made, now the transposon, the transposal element, whether it's been cut out of where it's at, or if it's been, generated anew, that's stuck in, and kind of one end of each bit of DNA is stuck to that transposon, that little bit of double-stranded DNA. And they can be as small as, 3,000 or 4,000 base pairs, right? So they can be really small. They can be really huge as well. So when that transposon is stuck in, now you've kind of rejoined the DNA, but you have these flanking pieces of single-stranded DNA. Right. And so those gaps are filled in by DNA polymerase, the kind of DNA repair mechanism. It goes, oh crap, single-stranded DNA, and comes in and fills in those two gaps. Now, what that means, because you had that overlap, and now it's like so, is that now you have what are called flanking direct repeats. Because if essentially the overlapping bit from the bottom strand is recreated, and the overlapping bit from the top strand is recreated. So every time a transposon goes in, it creates these repeats on either side. That's the characteristic footprint of a transposon. So you can actually find them simply by looking for, two direct repeats that are separated by, X amount of DNA. That's how we know that this much of our genome consists of transposons. So even if that transposon ups and leaves, it will leave behind this footprint, this flanking, these two repeats. That's what's called its footprint. So what do you think would happen if this transposon inserts itself into a protein coding region, the protein coding part of a gene? What would happen to the function of that gene? What do you think? It would shut it down. It would result in a non-functional protein being produced, right? Now, what about it if it went into the promoter region of a gene, the region of a gene that's used to control gene expression? What might happen now? There's a bunch of different possibilities, to be honest. Might do the same thing, might shut down transcription, right? So you effectively have the same issue, no functional protein. It might change transcription, right? So that it occurs in a different context, right? So actually, this is one of the mechanisms by which people think that transposons actually kind of fostered, change that was selected upon by natural selection, right? So the consequence of transposition right, very much depends on where this target sequence is and where the transposon goes, right? Because it's very disruptive. That's why Barbara McClintock discovered this, is because she was observing a transposon jumping into a pigment producing gene in maize. And so what she actually saw was a lack of pigment because a transposon had jumped in, maybe even jumped out again, but it still had left a chunk of DNA there that wasn't there originally. Right, so transposons are highly mutagenic. Now, if this bit of this transposition event is in, you know, one of the vast kind of wildernesses of our genome, where there's nothing of particular interest, no effect whatsoever, right? So that kind of shows you how, you know, these things can accumulate quite considerable numbers, because if they do so in regions where it's not, doesn't really have any consequence, then nothing's going to select against them. And so another characteristic, in addition to these flanking direct repeats and the transposase, are what are called terminal inverted repeats. Yes, highly mutagenic, right? Because wherever transposons insert themselves, Kiana, they're going to introduce a big chunk of DNA. And so if that's in the middle of a protein coding region or a gene, almost certainly it's going to change the function of that gene, usually for the worst. And so wherever they go, they're going to change the DNA sequence. What the consequences of that is depends on where it is they're transposing.
Does this process have like a checking process the way that the DNA replication thing?
Yeah, that's a good question. There are actually mechanisms which seek to inhibit transposition, right? So many eukaryotes have mechanisms that recognize transposase activity and try and shut it down. I don't actually know exactly how that works, to be honest. I'm not sure if it is known, but it's definitely something which is curtailed, right? Because it's generally not good. It's either not good or it's meh, neither here nor there for a cell, right? So if you happen to have a very active transposon, because they vary in their activity too, you'd want to kind of, you know, damp it down so it doesn't do a lot of damage, basically. So another thing of interest as well is you can get dead transposons, so they can die, right? Not in the they were alive and now they're dead sense, but if through just random mutation, either the transposase or these terminal inverted repeats accumulate a mutation, right? There's a mutation which changes their sequence. Quite often that stops that transposon from being able to move again. And so that's another reason why we have this, you know, nearly half of our genome consisting of transposons, is just through random chance they get inactivated. And so they just sit there, right? They don't do anything, right? They can't move anymore because they don't have a functional transposase, or they don't have inverted repeats which are recognized by the transposase. So that's another so when I was talking about like the evolutionary cruft in terms of our non-coding DNA, this is kind of one of the things I was referring to. All right, let's see what we got left. No, we don't have too much. We might get through this. We'll see. If we don't, we don't. It's not a big deal. So basically, there are two large kind of groups of transposons. There are DNA transposons right, which are kind of what I was talking about. Piggyback's one of those. And there are what are called retro transposons, right? And so retro basically means they go from DNA to RNA to DNA. And so that's why HIV is a retrovirus. Because it has a RNA genome that's made into DNA upon infection. And so retrotransposons, so they're not retroviruses, but they transpose via an RNA intermediate. And so the DNA transposons can either be replicative, right? So they copy themselves, or they can be non-replicative, which is what we call cut and paste. That's not in there. Cut and paste transposons. Do I have in the notes? No, I don't. So those non-replicative are kind of cut out and they're stuck somewhere else. Replicative stay where they're at and they get copied, and their copy gets stuck somewhere else instead. And retrotransposons, of which we have a whole ton of them, they're common in eukaryotes and they're also copied. And so that's one reason why their numbers have grown over evolutionary time, right? It's because every time they get copied, the count increases, and then you get more, and you get more, and you get more, right? And so unless there's anything to kind of damp that down or purge them from the genome, they'll just accumulate over time, essentially. And so these use a enzyme called reverse transcriptase. Right, and actually, that's an enzyme that I've used a whole bunch in turning RNA into DNA. Right, well, it doesn't turn it into DNA, it uses it to make DNA. Right, and that's a big part of that. We don't really have time to go into it right now because it's a very complex process. So I'm going to call it a day here, snarf some lunch, and see some of you in lab. So we'll finish this off on Thursday before we get into gene expression. All right, that's it for me. Have a lovely day if I don't see you later, and I'll see you on Thursday.